So I have mentioned basically all of these methods that we have in the facility. Now I also said that we run the cut and tag and the immune profiling. We also can do CRISPR screens together with another facility at KI. So it's a high throughput genome engineering facility. Yes. And I also want to mention that we recently bought a 10x chromium instrument and placed it in the biosafety level 3 in Biomedicum. So we can take samples that require higher biosafety level, for example, COVID samples. And I hope and I think we will see more of these samples. The services you've seen, the applications that we use is mostly 10x Genomics for 3' gene expression, for VDJ, for site-seq, and for CRISPR. We have been running SmartSeq 2 for several years, and during this last year, we are discontinuing and finishing all the SmartSeq 2 projects, and they were more than we expected. And now we are introducing SmartSeq 3 instead in the facility. All of the work that we do is with robotics, basically. Plate handling robots that can handle 384 well plates. Here we have a nanodrop, we have a mantis, and we have a TCAN as well. For the droplet-based systems, we use 10x Genomics. We also have another instrument from Takara, which is called Isolate Compact. And in this method, you can do limiting dilution and dispense cells in this chip over here to the right with the 5,000 wells. And we can build libraries that are similar to a SmartSeq library. And this method is good, especially for cells that are very large and cannot be FACS sorted, because there's no limit on the cell size here. So just like a quick outlook, I don't know if how my time is, but I think we will see more of these multimodal readouts that I've already mentioned. And I think we will see more methods for multiplexing your samples. And of course, a lot of projects will go to the different spatial techniques, looking at the spatial gene expression. And but for the single cell techniques that we work mostly with, I think we will see not more like an increased cell throughput, more improved quality and a reduced cost. I think that's where it's going. And finally, I just again want to point out how important it is to have good samples when you start your projects. I mean, most of you already have data, but now when you will analyze your data, you will I guess see how was the quality of the tissues, of the cells, of the nuclei that you started with. And if you have any questions about this, I think you can definitely contact us and we will try to help you to improve your protocols. And finally, if you want to contact us, maybe not running a project, but also discuss these issues of sample prep, you can find us on the SciLife Lab website. You can find us on the KI website. And if you want to look at the publications that we have contributed to, you can find them on the SciLife Lab website. You can find all the publications from all the facilities on this website. It's pretty cool, I think. But this is the link to our website.